So these are the different classes of apolipoproteins starting from A1, A2, A5, B100, B48, C1, C2, C3, ApoE and apolipoprotein A, right? So apolipoprotein A actually it's forming a part of ApoB100, right? ApoB100 ഒരു പാർട്ടാണ് apolipoprotein A, right? So that is it is found to be attached to ApoB100, and that is it cannot alone. We cannot see apolipoprotein alone on the surface of uh, lipoproteins. We can see the lipoprotein A or apolipoprotein A along with ApoB100, right? So it is a ApoB100. We can see it is on the major protein or lipoprotein holding the ApoB100 is LDL, right? That's why LDL is known as bad cholesterol. So wherever ApoB100 is there, or wherever LDL is there, there will be ApoB100. In majority cases, if the LDL value is found to be elevated, if the LDL, the lipoprotein, the concentration of LDL is high in a person, he'll be having large quantities of ApoB100 also with it. But along with ApoB100, there will also be a little rascal, that is apolipoprotein A, because apolipoprotein A is always found to be attached to ApoB100. It cannot be seen alone. It can be seen only along attached to ApoB100. And since ApoB100 is one of the major source of LDL, LDL behaves as a bad cholesterol, right? Because it is responsible for increasing the chances for plaque formation because it's having rich quantities of apolipoprotein A. And since it's a modulator of, or it impairs the fibrinolysis, right? So that is the significance of lipoprotein A, apolipoprotein A, this one, Apo A, right? Again, this A should be always written in the small letter, right? Okay, so next we move on to the metabolism of individual lipoproteins. So what are the functions we have already told, that is they are forming a part of the lipoproteins. They can act as cofactors like ApoA1, it's a cofactor of LCAT, etc. Then they also can act as ligands for the interaction with the receptors, right? That is ApoB100, then ApoE for LDL receptor, ApoA1 for the HDL receptor, etc. Right? Then next we move on to the protein metabolism of the individual lipoproteins, or how are these lipoproteins functioning in the body. We have already told that each lipoprotein has got different functions. For example, chylomicrons are responsible for transporting the exogenous TG, while LDL is responsible for transporting the endogenous TG, endogenously derived TG. Right? Again, HDL transports cholesterol esters from the extrahepatic tissues to the liver, while LDL transport the cholesterol esters from the liver to the peripheral tissues. Okay, LDL, then VLDL transport and endogenous TG, etc. So each lipoprotein has got individual functions to be carried out. So how are these molecules metabolized? How are they synthesized? How are they catabolized? How do they perform their function? That we are going to discuss, right? So first one is lipoprotein metabolism. The transport of first, for the metabolism to happen, they should be synthesized. So for their synthesis to be happening, what lipids should be provided first. Lipids in the transport lipid metabolism lipoproteins ഉണ്ടാവുന്നത്. So first the lipids has to be derived, right? And there are two sources of lipids. One is a dietary source, another one is a endogenous source, right? So first we'll discuss on the dietary sources. That is, when the diet is consumed, when a person is having diet rich in fats, what happens? The digestion happens. Digestion is followed by absorption, which happens in the intestinal lumen. And during the digestion, what happens? Lipases carries out the destruction or breakdown of the dietary lipids into cholesterol esters, free cholesterol, free fatty acids. Then TG, dietary TG is converted into DAG and MAG, right? And to a small extent, it will be completely broken up to give rise to glycerol and fatty acids. Now, what happens to all these dietary lipids? They will be absorbed into the intestinal lumen, where what happens? The TG and cholesterol esters has to be transported primarily because they are lipid insoluble in nature, while phospholipids and the free cholesterol are amphipathic. So these amphipathic molecule can be transported much comparatively easier when compared to that of the pure lipids like TG and cholesterol esters. So first the TG and cholesterol esters will be packed in the form of globules by this completely non-polar TG and cholesterol esters will be forming the inner core of the lipoprotein, and they will be surrounded by these amphipathic molecules like phospholipids and free cholesterol. Right? Yesterday we have seen the picture. And along with these phospholipids and these amphipathic molecules, the phospholipids and the free cholesterol, the different types of apoproteins also will be coming and getting assembled onto these molecules to form a lipoprotein. And the lipoprotein thus obtained or thus synthesized in the intestine is known as chylomicrons. So the apoprotein, the principal apoprotein which is found in the chylomicrons are the ApoB48. So during the absorption, what happens? These TG and cholesterol esters will be forming the innermost core of the lipoprotein, right? And that is followed by the enveloped portion, that is phospholipids, and the free cholesterol molecules envelops these TG and cholesterol esters. Along with these phospholipids and the free cholesterol, what happens? In between, there will be the ApoB48, which is produced, synthesized from ApoB100, which is synthesized in the liver. So ApoB100, which is synthesized in the liver, the same gene will be synthesizing the ApoB48, but in the intestine as a result of mRNA splicing. Right, so the same gene is present in the intestine also, but in the intestine, it is not the ApoB100 synthesized, it is only the ApoB48 synthesized. Clear? So gene is the same, right? In the liver, hepatic cells, gene and intestinal luminal cells, and the same gene and but the product is entirely different in the liver and in the intestine. In the intestine, the product is ApoB48, not the ApoB100, right? So the ApoB48 forms an integral part of this chylomicron. So that is how the chylomicrons are formed, and they get released out of the intestinal lumen, and they will be again chylomicrons. The chylomicrons are rich in TG. So instead of blood, they will be entering into the lymphatic vessels. Only a very small portion will be entering into the blood. The majority portion of chylomicrons will be entering into the lacteals, and they will be drained towards the thoracic duct, right? And in the thoracic duct, what happens? The chylomicrons will be stored. So the chylomicrons, majority of the chylomicrons will be taken up by or they will be entering into the lacteals and will be drained into the thoracic duct through the lymphatic system. And from the thoracic duct, they are taken into the bloodstream, right? And finally, they will be drained, or they will be taken towards the liver. And in the liver, what happens? The lipoprotein lipases are there, or hormone sensitive lipase are there. It is a lipoprotein lipase, and this lipoprotein lipase, the chylomicrons is having only a half life of 1 hour. 1 hour. After that, they will be entering into the catabolism, right? So the main site of the chylomicron metabolism is the adipose tissue and the skeletal muscles, because we know that the TG, the depot fat, are mainly stored in the adipose tissue and the skeletal muscles, right? So from the liver, what happens? Or in the liver, the chylomicrons will be acted upon by lipoprotein lipases, and they will be liberating the TG that is obtained from the diet, and the TGs will be entering into the hepatocytes. And from the hepatocytes, what happens? They will be taken up by they will be taken up by the LDL or the VLDL molecules towards the adipose tissue and the skeletal muscles. And again, on entering into the adipose tissue, what happens? Again, the lipoprotein lipase, which are present on the blood capillaries, acts upon these LDL molecules, breaks up the LDL molecules, and the TG contents will be deposited in the adipose tissue and skeletal muscles. So that's how the TGs are getting deposited in the body. So this is the transport of exogenous or the dietary lipids. So there are we have lipids coming from two different sources. One is the dietary lipids and other is the endogenous lipids. So that is the body itself can synthesize TG. We have already studied. So if the lipids are derived from the endogenous sources, that is, if body tissues are synthesizing the lipids, then it will be transported by the VLDL. So VLDL again mainly contains the TG and the cholesterol esters, which are surrounded by phospholipids and free cholesterol. And there the lipoprotein is ApoB100 and A1, rather than ApoB48, right? Then the transport, the absorption of the dietary lipids happens. Once absorption is happening, nascent chylomicron will be formed. Nascent means name, fresh. So nascent chylomicrons do not have what ApoA, E, and C, right? So nascent chylomicrons will be having only ApoB48 and ApoA. ApoA also will be there. Now C and E are derived from the HDL molecule. So during the absorption of the dietary lipids, when the chylomicrons are formed, for the first protein, of course, coming will be ApoB48. ApoB48 will be always followed by ApoA, the integral protein ApoA. So this is known as the nascent chylomicron. So nascent chylomicron contains the TG and the cholesterol esters inside, covered by the phospholipids and free cholesterol. Along with that, there will be rich quantities of ApoB48 and also ApoA. Such a chylomicron we call it as a nascent chylomicron. This nascent chylomicron then slowly enlarges its size with adding up the ApoA and ApoC and ApoE, which are derived from HDL, high density lipoprotein. And such a bigger molecule of chylomicron with additional attachment of ApoC and ApoE, we call it as the mature chylomicron. So the nascent, there's a difference between nascent chylomicron and mature chylomicron. Nascent chylomicron will be having the two apoproteins, ApoB48, which is present in rich quantities, and ApoA, right? And during the course of their journey from their intestine towards the liver, they further adds up ApoC and ApoE, which are donated by the HDL molecules. Such a chylomicron we call it as the mature chylomicron. So when a chylomicron molecule reaches the liver, it will be in the form of mature chylomicron. But immediately after getting liberated from the intestinal lumen, it will be a nascent chylomicron. So nascent chylomicron further adds more and more amounts of ApoC and ApoE derived from HDL molecule to give rise to mature chylomicron. And they will be moving towards the liver. And these chylomicrons, again, their course of their journey, they will be getting supplied. They will be supplying the TGs and the cholesterol esters towards the adipose tissue, the muscles, etc. Right? And these chylomicrons will be finally taken into the liver. Liver like what is happening? It will be supplying the lipoprotein lipase will be breaking up. It will be bringing within 1 hour. It will be undergoing catabolism, right? So during the course of the journey, what happens? Lipoprotein lipase can cause the destruction or can open up the chylomicron molecule and liberates the TG and the cholesterol esters from them, and will be these TGs and cholesterol esters will be getting deposited in the adipose tissues and the muscles. So what happens? Due to the reduction of quantity of TG and cholesterol esters, the chylomicrons can get reduced in their size, right? So the remnant of the chylomicrons will be found, and these remnants will be entering into the liver, right? So that is how the metabolism of chylomicrons take place. So during the metabolism of chylomicron, first digestion happens to the dietary lipids, after which the TGs and cholesterol esters will be formed. They will be undergoing absorption in the intestinal lumen, where it forms nascent chylomicron with the TG and cholesterol esters packed, surrounded by phospholipids and free cholesterol with ApoB48 and ApoE. So such a chylomicron we call it as a nascent chylomicron. This nascent chylomicron will be jumping out of the intestinal lumen and enters into the lacteals or the lymphatic system, taken to the thoracic duct, and during the course of their journey in the thoracic duct, what happens? They will be getting added up with the other apoproteins like ApoC and ApoE, right? And this ApoC and ApoE along with ApoB48 and ApoA, transporting the dietary TG and cholesterol esters, we call this a mature chylomicron. So it is having a larger size when compared to that of nascent chylomicron. Now this mature chylomicron move towards the skeletal muscles, the adipose tissue, etc., where it is acted upon by lipoprotein lipase enzyme, which causes the lipolysis of chylomicrons and liberates the contents in it. That is, the TG and the cholesterol esters will be liberated, or it will be donated to the adipose tissue and the skeletal muscles, so that the chylomicrons will be having now a reduced size. The size of the chylomicron will be reduced with a smaller concentration of TG and cholesterol esters. Now this small chylomicron molecule move. This is known as the chylomicron remnants. That is chylomicron itself. It's having TG and cholesterol esters with phospholipids, free cholesterol, ApoB48, C, A, E, etc. But in a smaller size, smaller size. It is known as chylomicron remnants. They will be moving towards the liver.